Good afternoon. I'd like to share some results from a DaCCoTA pilot study we did entitled Tissue and Genotyping Specific Expression of C-reactive Protein in the Oropharyngeal Tissue. Inflammation is becoming more and more recognized as a contributing, if not a causal factor, in many multifactorial chronic conditions, including cardiovascular disease and cancer. The venerable term inflammatory breast cancer demonstrates the long history of this recognition, for example. C-reactive protein, or CRP, is a key component of the innate immune system and clearly predictive of future CVD events, as well as a prognostic indicator in clinical cancer care. There's also considerable support for the concept of CRP as a causal factor in some instances, noting, for example, the increased incidence of colorectal and cervical cancer among those inheriting certain genotypes affecting circulating CRP levels. We've been exploring the possible involvement of CRP in the pathophysiology of preeclampsia at Turtle Mountain Community College for some years and proposed expansion of this work within the framework of the DaCCoTA study. To this end, we proposed a pilot study to enroll 20 individuals with known homozygous genotypes for RS-1205, which is known to influence serum CRP levels. Circulating CRP has classically been thought to be determined solely by hepatic production, but many other tissues have recently been found to express CRP as well, perhaps with primarily local effects though. Our aims were to determine if CRP expression occurs within the oropharyngeal tissue, if this expression is influenced by inherited RS-1205 genotype, and if we could demonstrate genotyping and expression results from formalin fixed tissue. We were able to enroll samples from 8 individuals, 4 of each homozygous genotype, before COVID pandemic essentially shut down recruitment. Samples were prepared and expression studies were done at TMCC with the help of our student research assistants. And you can see an example of one of our expression runs in the upper right corner here. We did demonstrate expression from oropharyngeal tissue, albeit at a low level of about 1,000th that of our housekeeping gene. Although severely limited by sample size, we obtained results showing a tendency toward higher expression among individuals homozygous for the RS-1205C allele, as you can see down in this lower area under results. The same direction of effect reported on serum levels. On the other hand, we also showed a similar trend toward higher circulating levels of CRP amongst those homozygous for the alternative allele. We were also able to demonstrate genotyping, but not expression results from formalin fixed samples. I'd be happy to entertain any questions.